Okay, this is a brief lecture on Mendelian inheritance and risk assessment. So in every cell in our body, we have, well, I have a nucleated cell. Inside the nucleus are the chromosomes. The chromosomes carry our DNA, which code for proteins, and that's the end product. Now, we have 23 chromosomes, 22 autosomes, and the sex chromosomes, and not all genes and all tissues are being active at the same time. So again, we have two copies of our genes, and the genes on there are known as alleles. So we have two alleles, except for the sex chromosomes. Genotype is our genetic material, like the genes that we have, the two copies. Phenotype is the observed characteristics, and it's based on the genotype and the environment. And what the environmental factors are, most of the time we don't know. Clinically, symptoms or features caused by a particular genotype are sometimes known and sometimes not. So a dominant disorder is if one of the two copies isn't functioning correctly and the individual exhibits the phenotype. Dominant disorders, we're going to talk about this as we move through the curriculum, but a lot of things that if only one of the two copies is abnormal, things with connective tissue are often dominant. Things with cardiomyopathy, as well as arrhythmias, a lot of the neuro stuff, one copy can cause a problem. The vast majority of disorders are recessive, which means both copies of the gene have to not be working. In most cases, one copy has one particular mutation and the other has a different mutation. So that's, there's two different types of abnormalities. We all carry genes that don't function correctly. Everybody carries about 6 to 8 genes that don't function correctly. But in recessive disorders, as long as we have one that does, we're okay. And every day we learn new things about what genes cause issues and, and so on and so forth. But again, we all are carriers for some genes that don't function correctly. The only risk is when both copies don't function correctly. If there's some disorder and you're taking an exam and you've never heard of it before, think recessive. It's some kind of enzyme thing, because like a lot of these enzymes, if you have one copy that's working right and it's an enzyme, 50% is usually more than enough. Usually 20% of most enzymes is more than enough that you don't have symptoms. So if it's something that you have no idea what it is, you read this thing and you have no idea what it is, think recessive. So now we're going to talk about recurrence risk. And that's, so if one parent is a carrier for recessive disorder and the other parent is a carrier for recessive disorder, both parents could give the normal allele. Mom could give the normal and dad could give the abnormal. Mom could give the abnormal and dad could give the abnormal, normal. Or both parents could give the abnormal allele. So when we look at a recessive disorder, there's a 1 in 4 chance, if both parents are carriers, that they're going to have an affected child. There's a 2 in 4 chance, or 50% chance, that the child will be a carrier, and a 1/4 chance they'll get 2 normal copies of the allele. So when trying to figure this out, we use something called Hardy-Weinberg, which is p squared, which is the normal allele, times 2pq, either, mom gave good and dad gave bad, or mom gave bad and dad gave good. So 2pq, that's a carrier, or q squared equals 1. It's most often used to determine frequency of heterozygotes or carriers of recessive disorders. So here, the genotype, again, are the genes that we have, and they're distributed proportionally. And in a population, they're usually in equilibrium. So if an allele A has a frequency of P and allele B has a frequency of Q, P plus Q equals 1. So population genotypes, AA equals P times P, or P squared. AB is P times Q plus Q times P, or 2PQ. And BB is Q squared, or P squared plus 2PQ plus Q squared equals 1. Now, there are a lot of assumptions of Hardy-Weinberg, which, quite frankly, are impossible to, meet. But anyway, there's assumption that there's no mutation occurring at the locus, that there's no selection for or against any genotype, that the population is large and mating is random, and there's no migration in or out of the population. So if all assumptions are met, you're at Hardy-Weinberg. So if the population is in equilibrium, changes to the allele frequency will average out in the next generation. Note, in reality, this is really hard to do, and other issues that can affect it include consanguinity and assortative mating. So, but let's just go through Hardy-Weinberg. So allele 1 is P and allele 2 is Q. P plus Q equals 1. P squared plus 2PQ plus Q squared equals 1. And the most important thing is that Q squared is the risk of the recessive disorder. So when they say something in the population is 1 in 50,000, they're talking about Q squared. If disease X has a frequency in the population of 1 in 100, then Q squared is 0.01 or Q is 1. P plus 0.1 equals 1, or P is 0.9. Of course, there's very few things that are recessive that have a population of 1 in 100. So in most cases, P is 1. Here it's 0.9, but 2 times 0.9 times 0.1 is 0.18. But again, in almost all situations, I'm just giving you an example of when we could actually look at P. P is basically 1, because if Q is 1 in 1,000 or 1 in 10,000, then this is 0.01 or 0.001, and P, just always think of P as 1. So, P squared is unaffected homozygotes. 2PQ is heterozygotes. In a dominant disorder, they'd actually be affected, and Q squared is affected homozygous. In a dominant disorder, this is almost non-significant. So, if you're looking at a dominant disorder, 2PQ is actually the frequency in the population of affected heterozygotes. In comparison, in a recessive disorder, P squared is unaffected homozygotes, 2PQ is carrier heterozygotes, and the affected homozygotes is Q squared. And again, 2PQ is really 2Q. So you take the square root of Q and you multiply it times 2, and you get the carrier frequency. PKU, phenylketonuria, is a metabolic disorder, which we'll probably be studying when we get to vital fluids, fluid to fuel. PKU is a recessive disorder with a carrier frequency of 1 in 4,500 in Ireland. If all disease-causing alleles are grouped and treated as a single allele, the frequency of Q, therefore, is 1 in Q squared is 1 in 4,500. So, and again, when you're taking an exam, if the number is super big, you assume that that's the carrier frequency of a recessive disorder. So Q squared is 1 in 4,500. Q is the square root of that. And again, P is basically 1. So 2 times Q is about 0.03, about 3%. So about 3% of the population in Ireland is a carrier for PKU. So let's go through this. The incidence of Huntington's disease, which is a dominant, is 1 in 10,000. What is the frequency of the HD allele? Again, now we're talking about a dominant disorder. 2PQ is 1 in 10,000. So Q is is double that, or 1 in 20,000. So that's the frequency. So let's look at dominant inheritance. When we're looking at something dominant, we see it in many generations. We're going to go through this more and more, which is a recessive disorder, the first person in the family that has a disorder. So Most disorders, as I said, are recessive. We all carry bad genes, and the phenotype is only seen when we have two individuals. So we usually don't see it in the family history. We only see in fact, the only people that are usually also affected is in the same generation and usually within the same family, the two parents. So if a child has a recessive disorder, there's a 25% chance of having another one. And this is just every single ethnic group is at risk of different things. And this is when in genetics, I always ask, what's your ethnic background? If you're African American, I'm worried about sickle cell. If you're Caucasian, I'm worried about cystic fibrosis. If you're from the Mediterranean, I'm worried about thalassemia. French Canadians or Ashkenazi Jews, I'm worried about Tay-Sachs. So every ethnic group, there's a different thing that I'm worried about. A Caucasian woman is pregnant. Her spouse has a brother with cystic fibrosis. The husband is not affected with cystic fibrosis. What is the chance that they will have an affected baby? Okay, so this is, whether I call it cystic fibrosis or any other kind of recessive disorder. So the first thing, if the brother is affected with a recessive disorder, then both of his parents have to be carriers. So what's the chance that the husband, the person that we're worried about, is a carrier? We know he doesn't have cystic fibrosis, so looking at our Punnett square, which I showed you earlier, there's only three other possibilities. One is he got two good alleles. Mom gave good and dad gave bad. Mom gave bad and dad gave good. So there's only three possibilities, and two of the three, he's a carrier, okay? So whenever you see, I'm going to go back to this, whenever you see this kind of pedigree, that they're wondering about the risk of somebody who has a sibling that has a recessive disorder, this number is going to be 2/3. So there's a 2/3 chance that dad's a carrier. What do we use for moms? There's no family history of cystic fibrosis, so we use the population risk. And they would have to give this to you unless I gave you a document that showed what the risk is. So they'd have to give you the population carrier frequency for this particular disorder is 1 in 25. What's the chance that they both give the bad gene? Well, 1 in 4, right? So 2/3 chance that dad's a carrier times the carrier frequency for mom, because there's no family history, times what's the chance that they both give the bad allele, and that's 1 in 4. So there's 1 in 150 risks of having a baby with cystic fibrosis. This is the question, however they call it CF, they call it PKU, they call it any kind of recessive disorder. This is the exact question, and if you forget about this 2/3 here, you're going to get it wrong, okay? So they're going to ask the same question over and over again, and that's how you get it right. So I took this out of a USMLE book, and sorry that the 1/4 looks weird. It's just when I did the question on my PowerPoint. A condition shown in the family history is known to have a prevalence of 1 in 40,000 in the general population. Assuming the most likely interpretation of the pedigree shown, what is the probability that the first daughter of the affected male will be affected? So let's look at this. We have, well, they're all males that are affected. There's almost nothing that's Y-linked, okay? But if it was Y-linked, what would the chances his affected daughter be affected? Well, the answer would be 0, but 0's not here. So what's the other possibility that we see something in many generations? Autosomal dominant. If he has an autosomal dominant disorder, what's the chance that he will have an affected child? And the correct answer is 1/2. So I actually, like I said, I took this out of a USMLE test question book, and again, if you were looking at this and you're like, oh, it must be Y-linked because there's all males affected, and that's not even an answer. So there's almost nothing that's Y-linked. So if they're asking about his daughter, if they asked about the son, you might have the risk of putting 1/2, but he asked about the daughter. Anyway, that completes this lecture.